Hello, and welcome to the Analytical Services Lab at the Complex Carbohydrate Research Center in Athens, Georgia. My name is John, and I am a research technician here. Today, we will be going through the procedure for glycosyl composition using Trimethylsilyl, or TMS. This method allows us to break down oligosaccharides and polysaccharides to detect monosaccharides, including amino sugars. This method involves 3 steps. First, a methanolysis, which is a hydrolysis done in methanolic conditions, which breaks the sugar chains into monosaccharides. For amino sugars, the acetyl groups come off the amino functionalities, making them NH2. Secondly, a re-N-acetylation. We use methanolic conditions to acetylate the nitrogen bonds in amino sugars. We only want N-acetylation, so we use low temperature and methanol. Lastly is the trisilylation, where we replace the OH groups with O-TMS groups to allow for analysis on the GCMS. Here we see a starch molecule going through the 3 steps of the TMS derivatization. First is methanolysis, which breaks up the polysaccharide into monosaccharides. The re-N-acetylation does not affect the molecule here due to not being an amino sugar. And finally, the trisilylation, which replaces the OH groups with O-TMS groups. Here we have the glucanac disaccharide in the beta-alpha form. In the first step of methanolysis, the bond between the 2 glucanacs are broken and replaced with an O-methyl. The acetyl group, which was attached to the amino sugar, has now become NH2. The next step, the re-N-acetylation, adds that acetyl group back onto the nitrogen. And finally, the trisilylation changes the OH groups into O-TMS groups for analysis. Before we get started with the experiment, remember to always follow lab safety guidelines and wear a lab coat, gloves, and eye protection. We will be running a sample that has been lyophilized or freeze-dried to remove all water. 20 micrograms of inositol is added to the sample prior to drying to serve as an internal standard in our analysis. With our sample, we run 2 sets of standards. The 2 standards are because some sugars elute at close times, and for quantitative analysis, we need good separation. We use 50 micrograms of each standard and 20 micrograms of inositol as an internal standard. Here you see the 2 sets of standards and the sugars included in each. Now to begin the first step of the experiment, the methanolysis. After the sample is completely dried, add 1 molar of methanolic hydrogen chloride to your sample. We add 400 microliters of this methanolic HCl solution and place the sample on 80 degrees Celsius for 16 to 18 hours. After 16 to 18 hours on the heating block, the samples are dried under a nitrogen stream. 400 microliters of methanol is added 2 separate times and dried off to ensure that all the hydrogen chloride acid has been evaporated. Visually, the sample should appear to have a white residue dried on the edge of the tube walls to indicate that the hydrogen chloride has been evaporated. Next, we move on to the re-N-acetylation. We add 200 microliters of methanol, 100 microliters of pyridine, and 100 microliters of acetic anhydride. The 2:1:1 ratio is important. We vortex the solution and allow this to sit at room temperature for 30 minutes. After sitting at room temperature for 30 minutes, the samples are then dried down completely under nitrogen. This takes about 30 minutes. After our samples are completely dry, we add 200 microliters of our TMS reagent, or Trisil, which is used to volatilize them and make them O-TMS residues. This is then put on a 80 degrees Celsius heating block for 30 minutes. After heating for 30 minutes, the sample is then taken off and dried down under nitrogen stream almost completely. It is important to be careful to not over dry your sample or you could lose it because the sample will now be volatile. Next, 500 microliters of hexane is added to the sample to reconstitute it. The sample is vortexed and centrifuged to pellet the leftover TMS reagent. The hexane layer is then transferred to another tube to be dried down under nitrogen stream to a level of about 150 to 200 microliters of hexane, which is then transferred to a GC vial insert for injection. Now we will be looking at the total ion chromatograms for our standards and our samples. First, let's look at our standard 1. In standard 1, we have arabinose, ribose, fucose, galactouronic acid, glucose, N-acetylmanosamine, and N-acetylglucosamine, and finally our inositol internal standard. In our standard 2, we have rhamnose, xylose, glucuronic acid, mannose, galactose, N-acetylgalactosamine, and our internal standard inositol. Notice how in this standard 2, galactose has 4 peaks which we integrated and identified, while mannose only has 1. Each sugar residue can have up to 4 peaks to indicate the alpha, beta, and pyranose and furanose forms of each sugar. For quantitation purposes of our standards and sample, we only integrate the most major as well as consistent peaks for each sugar residue. In this example, our sample, which is mainly arabinoxylane, shows arabinose, 1 identified residue of rhamnose, fucose, xylose, mannose, galactose, glucose, and our internal standard inositol. In order to calculate the mass of a sugar residue in our sample, we have to calculate a response factor value using the standard. We will be calculating the response factor value for xylose. We have taken the integration results from standard 2 with xylose and inositol residues. At the bottom of this slide, you can see the calculation that gives us a response factor value of 0.614. Now we need to integrate the xylose and inositol peaks in our GC chromatogram example. Here we have the integration results and have highlighted which peaks we are looking at for our response factor equation. Now we will use the response factor value equation in order to calculate the mass of xylose in the sample. We have the response factor of xylose from our previous calculation using the standard, as well as the area of xylose residue and area of inositol from integrating our sample. We also have the mass of inositol that was used in the sample. We are able now to fill out the equation and solve for the mass of xylose residue, which is 63.3 micrograms. We can calculate an RF value for each sugar found in the sample, and knowing how much sample was originally used, we can come up with a percentage for each sugar, as well as summing all the sugar masses in order to find a total carbohydrate percentage by weight. Let's go over 2 frequently asked questions during our courses. First, these beads at the bottom of this methanol chemical bottle are known as molecular sieves. These molecular sieves are used to dry the solvent. Water will interfere with our methanolysis, so reagents must be dried using these sieves. Second is our nitrogen blower setup. We use the University of Georgia's Fabrication Center to create custom nitrogen blowers, which feeds nitrogen from a tank through glass pipettes to dry things down.